this video we will discuss about which region of this Smad3 is critical for cell motility in the TGF-β induced cells. In the previous video we have find out that the this end terminal of the Smad3s are critical, but which regions of this Smad3 plays a role towards cell motility, that we have not find out. And from the heading it's clear that the beta 4 region of the Smad3 is critical for cell motility. So how they come up to this point, we will discuss that experimental results in this figure. So in order to find out which region of this Smad3 plays a role towards this cell motility in TGF-β induced cells, what it is, they prepared the knockout at different regions, that is shown here. That they prepared the knockout for this alpha 3 regions, alpha 4 regions, beta 4 regions, and the point at which they did the mutations are shown here. So that is N3133 mutations, N3311, N3311A, and 3311B. So these are the mutations that are done, and in these mutations, this amino acids are being replaced by their corresponding amino acid that are found in the Smad1. Thereafter, they check the phosphorylations of the C terminals of the Smad3 in those when these mutated Smad3 are infected into the Smad3 knockout A59 cell lines, and we can see that the phosphorylations of the Smad3 are shown when these lentivirus are infected. These lentivirus infected these cells. So this indicates that the Smad3 phosphorylations pathways get triggered, and the lentivirus infections also works well. Thereafter, they check the different activities. First, they check the KAGA activities using the luciferase reporter, and here all the infected cell lines, different mutated infected cell lines show KAGA signals. And when they check for this morphology change, they can see that the morphology change also occurs for all the three cell lines, and they are comparable to the wild type A59 cell lines. After that, they check the E-cadherin levels when the cells are induced with TGF-β for 24 hours. And they can see that E-cadherin level goes down for the control case, but not for the case where Smad3 is knocked down. But when the Smad3 is infected, either wild type Smad3 is infected, they can see a decrease in the E-cadherin signals, and for all the different three mutations also they see a decrease in the E-cadherin signals. Sorry about it. Yeah, they see a decrease in the E-cadherin signals. That can be seen here, and the decrease in E-cadherins are highest for the N331A. And when they check the stress fibrin formations, they can see that stress fibrin formations are also occurred in all the three infected cell lines. So far, all of them shows a positive results for the cell motility, all the mutated cell lines. And when they check this, and this is the immunofluorescence image of the E-cadherins, and this is just the image of the Western blot that is done in the panel E. So these two are the different techniques but shows the same results. That is, E-cadherin signals goes down for all the three mutated cell lines, and for all the three mutated cell lines, and also for this wild type A549 for this wild type A549 cell lines. But right, and that can be seen here, that is N3133A, we can see the signals are lower when the TGF-β is induced, and similarly for N3331A and 3331B. And when they check the migrations or the cell motility in different cell lines, what they are able to find out that the wild type cell line shows an increase in the migrations or cell motility, and when the knockout cell lines are induced with the wild type Smad3, they also shows the increase in the migrations. And when they use the N3133A, it also shows the migrations. And when they use N3331B, mutated cell lines, mutated infected cell lines, they also shows the migrations. But when N3331A is infected into the Smad3 knockout cell lines, they do not see increase in the migrations. So this might indicates that it's the beta 4 region, as this N3331A mutations occurs in the beta 4 region. So it's the beta 4 regions that plays a role towards these cell migrations. This is how they come to a conclusions that beta 4 region of the Smad3 are critical for cell motility in a TGF-β induced cells. So I hope now you have understood the what are these beta 4 regions, how these beta 4 regions of the Smad3 is how researchers come to a point that beta 4 region of the Smad3 is critical for cell motility in a TGF-β induced cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.